Okay, everyone. Good morning or good afternoon, depending on where you're joining us from. I'd like to get started so we can get this webinar rolling today. So on behalf of Biocytogen, I'd like to welcome everyone to today's webinar. We're very excited to invite back some of our antibody discovery experts that will provide some insights to the capability of our platforms. So before we continue, I'll briefly introduce myself. My name is Jenna Frame, and I'm from the marketing team here at Biocytogen. My scientific expertise spans the use of animal models in the fields of hematology, oncology, and immunology. Our panelists for today's presentation include Dr. Toru Kanke, CEO of LiberoThera, who has been leading the company since 2020. Toru has over 25 years of experience in pharmacology research at industry, academic, and biotech companies, with expertise in cell signaling, high-throughput screening, and antibody drug development. He obtained his bachelor's and master's degrees from the University of Tokyo and his Ph.D. from the University of Strathclyde in the UK. Thanks very much, Dr. Kanke, for joining us today for the Q&A portion of today's presentation. Next, I'd like to briefly introduce today's speaker, Dr. Yanan Guo. Dr. Guo received her Ph.D. in neuroscience and pharmacology from the University of Rochester and worked as a postdoc fellow in translational medicine at Brigham and Women's Hospital at Harvard Medical School. In 2014, she joined Biocytogen and served as the director of pharmacology. Since 2020, she served as director of antibody therapeutics. Dr. Guo has over 10 years of experience in cancer research with broad experience in drug screening, pharmacologic and toxicology analysis, and the development of new antibody drugs. Dr. Guo is joining us live from our Beijing site, and I'd like to thank her for her time late at night and invite her to begin the presentation without any further delay.
Okay. Thank you, Jenna, for the nice introduction, and welcome everyone to today's webinar. And today I'm going to talk about how to overcome the difficulties in GPCR antibody discovery using our Renmice-Based platform. So today's talk contains three parts. First, I will briefly introduce our company and our fully human Renmice platform, which is the foundation of all antibody discoveries that we have been working at Biocytogen. Then I will show you our established GPCR platform, and which can enable the efficient antibody discovery for GPCR targets. Then finally, I will focus on one of the leading CCR8 antibody discovery program that we collaborated with LiberoThera. So Biocytogen was founded in 2009, and now we are a clinical stage global biotech company. And we have a headquarters at Beijing in China. And also we have sites at Jiangsu, Haimen, Shanghai, Boston in the US, and also the offices at San Francisco, and also at the Heidelberg in Germany. And we have different innovative antibody discovery platforms, including the fully human Renmice platform, and also the bispecific ADC platforms using the antibodies generated from the common light chain light mice. And also we have a TCR mimic platform, which can generate antibodies recognizing the intracellular tumor antigens. And finally, we have a GPCR platform which will be the major focus of today's talk. And for the pipelines, we have 10 core assets with the first-in-class and best-in-class potential, and among of them, 5 of them are already in clinical stage trials. And we also initiated a large scale project to develop the antibody drugs for over 1,000 targets. That program called Project Integrim, which was launched in 2020, and now we have over 700 targets already have their antibody hits. And now we, based on all these assets and platforms, we have established partnerships with our collaborators, and also out-licensing some of our assets and platforms. So at Biocytogen, we can make antibody drugs in different drug modalities, which including the monoclonal antibody, the bispecific antibody that generated from the common light chain mice, and also we can conjugate the linker and payload to that bispecific antibody to further develop bispecific ADC drugs. So those drugs can increase the antibody binding specificity only to the double positive tumor cells, and also reduce the toxicity to the normal tissues. And we also can develop nanobodies using our Renmice. And the nanobody has smaller size, can penetrate into the tumor tissues. So that has many advantages. And we also, as I mentioned, we also have a T-cell mimic antibody can recognizing the intracellular tumor antigens that presented by the MHC molecules as a complex. And we also have a GPCR antibody discovery platform. So to develop the drugs in different modality, we established the different antibody discovery platforms. So we have the Renmice mice, which can generate monoclonal antibodies, which is fully human, and also to generate more antibodies with diverse sequences, germline gene usage, and epitopes. We choose to knock out the target gene first. Then the antibody generated from those target knockout mice will recognize more conserved region between different species, especially for those genes with high homology between human and mouse. We can generate antibodies recognizing different species using the knockout mice. Then we also have a common light chain Renmice mice, which can generate antibodies sharing the same light chain sequence. So that will help the assembly of the bispecific antibody easily. And also you can generate high purity and good developability bispecific drugs. Then conjugating the linker and payload, you will generate the bispecific ADC drugs. And as I mentioned earlier, we can generate nanobodies, the T-cell mimic antibodies, and as well as GPCR antibodies. So the GPCR antibody here, again, we use the Renmice platform. The Renmice platform actually have 3 different mouse models. The Renmice, which can generate monoclonal antibody, the Renlight can generate a common light chain antibody, and the Rennano can generate nanobodies. So all 3 together, we call that Renmice platform. The Ren means human in Chinese. So the Renmice platform can generate antibody with fully human sequences. Again, we use Renmice and also knock out the target gene first. Then we will be able to generate more antibodies recognizing the conserved functional regions. So that's our platform. So back to the Renmap. I just want to show you how we generate that mice. We use a special gene editing technology that developed by ourselves. So we can do a mega-base scale gene editing. So to replace the entire variable region of the antibody heavy chain as well as the light chain with the human counterparts. Then those mice can get robust immune response compared to wild-type mice. And also we see the antibodies generated from those mice can have very high binding affinity, even at some nanomolar range. And those antibodies, since we replace the variable region, so those antibody generated using the fully human sequences for the variable region of the antibody sequences. So after 1 step switch to the human constant region, you will get 100% human antibody. But for wild-type mice, even after the switch of the constant region and after the humanization step, you will finally end up with an antibody with up to 90% of human sequences. So compared to that, the fully human antibody will have lower chance to generate ADA in the human bodies. And that will finally lead to the better efficacy and also the higher successful rate during the clinical stage development. But to generate antibody, the most difficult thing is that some proteins has high homology between human and mice. So here we analyze 150 targets. So as you can see, majority of them showing the sequence homology over 50%, but some of them even higher 85%. For example, the CD73, you can see the sequence homology between human and mouse is 90%. So if you use wild-type mice for the immunization, you will only get the antibodies recognizing those non-homologous regions, that 10% of the difference sequences. But for the conserved region, it's very difficult to generate the immune response due to the immune tolerance in the mice. So our strategy is that we will knock out the mouse gene first. Then the mouse no longer seeing the same gene at all. Then immunize with the human antigens. You will end up with the sequences with more diverse sequences and germline gene usages, as well as the epitopes that even recognizing the conserved regions between human and mouse. So here just showing an example that we actually did before. So we tried different drug targets. Here we have 5 different targets. We use either the Renmice mice or the Renlight mice. We see in either mice using the target knockout, we will generate a lot more antigen positive clones compared to the wild-type mice. And also all the protein sequences between human and mouse is over 50%. So that means those targets is relatively not easy to generate antibodies. But if you knock out the target gene, it will help to increase the chance you get more positive clones. Then for the screening part, we use a system called Beacon and from the Berkeley Light, which is a single B-cell high throughput screening system. So using that system, we can run 4 chips at the same time. So each chip has 14,400 pens, the little pens here. And you will see the machine can load each single plasma B cells into the little pen. And then the B cells, if the B cell screening the antibodies, you will see it will be light up by the labeled antigens above the wells like this. So using that machine, we can quickly identify the plasma B cells which can secreting the functional antibodies. So that step's very quick. We can do that within 1 day. So that will save you a lot of time for antibody discovery. And this machine also do the antibody screening in a high throughput way. So you can see that we can screen more than 50,000 single B cells within 1 day. Okay, so as I mentioned that we using that Renmice platform, we knock out the target gene first, and we also using the Beacon high throughput screening machine. So we did a large scale high throughput antibody discovery. So for the over 1,000 drug targets. So here is the graph showing that we actually immunize the knockout mice with the antigens, try to generate the antibodies with novel epitopes, and some of them even recognizing different species, and those recognizing both the non-homologous regions as well as the conserved region. And all the antibodies will further run the in vitro assays, and also we have the capability to do the CMC development. And also after running with the PK, PD, and tox studies, they will move into the clinical stage. So that's the whole process of the project Integrim. So here is the progress of this project Integrim. So as you can see, we have established 50 partnerships for our assets that developed from the project Integrim. And by ourselves, also developed more than 30 PCC molecules. And also we have antibody hits available for over 700 drug targets. So that's a large scale project, and we are currently working on, and we already generate a lot of antibody hits that can be chosen from, and also for out licensing. And those antibodies may cross with different species, which is convenient for your in vivo efficacy studies and the further tox studies in monkeys. And also using that platform that can increase the chance for us to obtain the antibody recognizing the difficult target like GPCR. So then here I'm showing just some representative early stage binders that we generated from the project Intergram. Majority of them are in the oncology field and autoimmune disease field. And we have also some drug antibody hits in other fields like neurology, metabolism, cardiovascular, and infectious disease. This is not a complete list, just a very short example. So if you are interested in any of them, you can check the information on our website or contact our BD team for further information. So then let's move to our GPCR platform. So we all know that GPCR is the most difficult target to generate antibodies. But in the entire human genome, we have over 800 GPCR proteins, which are very important for the many functions like neurologic functions, the gastrointestinal functions, immuno-oncology, and others. Among those proteins, about 370 GPCR proteins are druggable targets, and some of them already approved. Some drugs already approved, like CCR4, CCR5, and CGRPR. But in the past 50 years, only 1/4 of them have been generated drugs, but the rest of them haven't been touched yet. And we know that the majority of drug modalities for GPCR are small molecules, but antibody drugs only account for less than 2%. That because it's very difficult to generate the GPCR protein in its native structure as an antigen for the immunization. That's the first difficulty. Then some GPCR proteins has very limited extracellular regions, which will not induce enough immune response. And that's why people are trying hard to generate antibodies, but not always successful. So that's why we established our own GPCR discovery platform. First of all, we have the fully human Renmice platform, and we knock out the target gene first, and then we use a high throughput beacon screening system. So combining all this together, we were able to increase the chance to generate more antibodies against GPCR proteins, and even recognizing the conserved functional regions. And some antibodies can recognize different species, which is convenient for the downstream screening and safety assessment. And also we optimize the immunization methods. So I will show you more information in my following slides. So first of all, if you look at the GPCR family in the human bodies, there are 5 major GPCR classes. The family A, which is the largest GPCR family, including the chemokine receptors, and also that's the largest group of targets for existing drugs. And the family A GPCR proteins has very, very short N-terminus regions, and also very limited regions for the other 3 extracellular loops. So that's the most difficult targets to generate immune response. And then you see there are several, there are other classes of GPCR proteins like the family B, for example, the GLP-1R, which can activated by the peptide hormones and regulate the homeostatic functions. And the family C play important role in the CNS and calcium homeostasis. And the family F involved in the tissue homeostasis and also the adherent GPCR. So if you look at the picture, it's clear showing that only the family A showing very short N-terminus regions, but the others have pretty relatively longer N-terminus regions, and some even can form a dimer. So if you're trying to generate antibodies recognizing those proteins, it's a little bit easier to generate antibodies targeting those type of GPCR. So here is showing antibody drug that currently in clinical trials or have been approved. So I can see here is a tree showing that some drugs have already approved, like the CGRPR, CCR4, and CCR5. And also there are some others like CCR7, CCR8, CCR2, C5AR1, CB1, GLP1R, and others are currently in early clinical stage. And people are also trying to generate ADC drugs targeting the GPCRs that can be used as a TAA. So as you can see, here is a table showing that the drugs that target the GPCR as a TA, for example, the CXCR4, the LGR5, GPR56, CCR7, GPR20. So majority of them are currently in the class A, the GPCR family, and all of them are currently in very early clinical stage in the preclinical or phase 1. So our platform is first of all, we use the gene target gene knockout mice. So if you use wild-type mice, you will generate antibody recognizing the epitopes at the different between human and mouse. And if you knock out the target gene, you will generate antibodies recognizing more epitopes, and some of even recognizing the conserved region between human and mouse. So that's the first strategy. Then we optimize the DNA immunization methods. First of all, we are trying to use the DNA method immunization as a preferred method because the DNA plasmid can be delivered into the mouse and also generate the proteins in its native conformation. And also that expression is a kind of long-term. And we also optimize the peptide that fills at the C-terminal region of the GPCR, which can strongly bind to the MHC molecules and then enhance the immune response by activating the CD4 T cells. So the DNA immunization is the preferred method we would like to use first. But besides that, we also trying different immunization antigens. For example, using mRNA to deliver a GPCR gene, but that one may have a shorter storage time, but it can also form a natural GPCR protein with a rapid expression. And also we try the overexpressed cell lines and also the virus-like particles, and these two can also form natural conformation of GPCR proteins, but some of them can generate non-specific antibodies, and the VLP is kind of difficult to formulate. And also collaborating with LiberoThera, they have a special technology can make the proteolipids, and also the antigens can be presented as a natural conformation, but again, it's difficult to formulate. So among all these methods, we prefer to use the DNA immunization first, but for some very difficult targets, we also try different methods, the combination of different methods. So after the immunization, we'll use the beacon machine as well as other drug screening methods like phage display or even hybridoma. Then we identify those positive binders and get the sequences and to test the in vivo and in vivo functions, in vitro and in vivo functions. Here is the progress of our GPCR platform. So we have more than 80 GPCR targets being investigated in the Renmice platform, covering most classes of GPCR proteins. And as mentioned earlier, the largest class is the class A. And among them, about 47 of them, we already get antibody hits, and about 22% of them are still not started yet. But still, we know that there are about 24% of them, even though we try different methods, we haven't got any serum titers. So that probably because that's the most difficult targets with very limited extracellular regions, and then we need to put more effort to overcome the difficulties. So here we just summarized our GPCR platform. We are using the fully human Renmice platform with the target gene knockout and also the beacon screening system. We obtain antibodies with more diverse and novel epitopes, and we recognize different species. And we also optimize the immunization methods. And here is the partial list of the antibodies we have already generated for these GPCR targets. There are some of them very known targets, like CCR8, GPRC50, CCR259, GLP1, and others. And then I will focus on our leading GPCR program that we collaborated with LiberoThera for the CCR8. So for this project, we develop a fully human antibody targeting CCR8, and we show that antibody have a good in vitro and in vivo efficacy results. So just a little background about CCR8. CCR8 is specifically expressed at high density on the cell surface of many human tumor infiltrating Tregs. As you can see, compared to the normal colon and healthy PBMC, all the other tumor tissues showing high expression of Tregs, and also the CCR8 is highly expressed only in the Tregs, the tumor-infiltrated Tregs, but not in the conventional Tregs or T cells or the CD4 positive T cells. And if you look at the expression pattern, CCR8 is the most similar one to the well-known Treg biomarker, the FOXP3. So that suggests CCR8 is primarily restricted to the Treg cells. So that makes this a very important and attractive target for the Treg depletion. So we started this collaboration with LiberoThera about 2 years ago, and at that time I actually tried different immunization method. We actually use DNA. We actually use VLP, the commercial available one, and also LiberoThera provided their proteolipids for CCR8. But for that round of immunization, the DNA and VLP didn't work well. But the proteolipids generate a strong immune response in all the mice that we immunized. So again, we use the mouse CCR8 gene knockout mice for the immunization. Then we use different screening methods, including the hybridoma, phage display, and the beacon. With all these methods, we were able to generate the antibodies that with good in vivo and in vitro functions. So here is the competitive landscape. Our clones have human IgG1 isoform with ADCC enhanced functions, and also we can see that the benchmark for the CCR8 antibody actually from the Shionogi, from the Gilead and BMS, even the Lanova, and they are currently in the clinical stage 102. But all these antibodies can only recognize in human CCR8. However, our antibody can recognize human, monkey, as well as mouse CCR8. And for the mechanism, they are quite similar. We all develop a antagonist antibody, which can block the binding of the CCR8 and with its ligand CCR1, and all of us choose the ADCC enhanced FC. So here, just want to show you some data. Our clone A, B, and C actually showing in different colors. And the benchmarks from Shionagi and Gilead are shown here as gray, black or gray colors. So all these clones can combine to human CCR8 pretty well. And our clones can recognizing the mouse CCR8 and the monkey CCR8. However, all the benchmarks only recognizing human, but not to the other species. And if you look at the binding specificity of all these clones to the CCR family members, we here tested the binding of those to the overexpressed CHOA cells. As you can see, all three clones only recognizing CCR8, but not to other family members like CCR2, 4, 5, and even the CX3CR1. So that means our antibodies are very specific for CCR8. And then we use a calcium release assay to look at the antagonist activity of all these clones. So again, all our clones showing a better, a comparable or even better inhibition of the calcium release compared to the benchmark controls. And also we did a protein engineering on the FC region to enhance the ADCC function. Here we use actually a Promiga kit that can evaluate the ADCC function. So once the antibodies bind to its target cells expressing CCR8, it will bind to the effector cells and then turn on the luciferase expression. So by marrying the luciferase values, you can see the ADCC function. So here all our clones showing a similar ADCC activity compared to benchmarks. And we all know that the Treg depletion requires the ADCC function. So the ADCC enhancement can actually improve its T cell expression, T cell depletion. So here we compare the original clones using the wild-type IgG1 isoform to our enhanced clones with the protein engineering. So the enhanced ADCC enhanced clones are actually showing shown by the solid lines, and compared to the dotted lines in the wild-type FC, you can see the protein engineering can enhance the ADCC function for all clones, clone A, B, and C, and even for the benchmark antibodies. Then, based on all these in vitro functions, we think these three clones are pretty good. Then we move on to the in vivo efficacy test. So for the in vivo study, we use CCR8 knock-in mice. So by using these mice, we can see all the ADCC enhanced clones, our clone A, as well as the benchmark antibodies showing better in vivo efficacy than the original clones that with the wild-type IgG1. And our clone actually showing a comparable in vivo efficacy compared to the other two benchmarks as a single drug. And if you do the combination of the combination treatment using our CCR8 antibody together with the PD-1 analog in the triple humanized mice, you can see there is a synergistic effect when you're combining those two, as shown by the purple line at the bottom. So suggesting that the CCR8 can either be used as a single drug or used as a combination with either the PD-1 or PD-L1 drugs to further enhance the in vivo efficacy. For the other two clones, we see pretty similar results. For the clone B, as a single drug, showing comparable in vivo efficacy results. And for the combination here, this time we only showing a similar results as a single drug or a little additive effect. For the clone C, also showing good in vivo efficacy and a synergistic effect in combining that drug with the PD-1. So we then look at the physical chemical properties of all these three clones. They all showed pretty good results, and also they are pretty stable under different stress conditions, either as 40-degree or low pH at pH 3.5, or the strong deamination conditions. Under either conditions, they are pretty stable. So just in summary, so we actually developed a novel ADCC enhanced fully human antibody against CCR8. And those antibodies have multiple species cross-reactivity, which can recognize in human, mouse, and monkey, and a high binding specificity only to CCR8, but not to other CCR family members, and strong ligand binding blocking activity, and also strong in vivo efficacy as either a single drug or using as a combination with the PD-1 drug in the mouse models. So those antibodies, we will submit the patent by the end of this year. And also the global rights of all these antibodies are open for licensing. So if you are interested in any of them, you can contact us. Yeah, just the final slide. So I just want to show you that with all the technology platforms that I have shown in my slides, and all the antibody assets, either the earliest assets from Project Integrim or our PCC stage assets, and even some of the clinical pipeline assets, we are happy and welcome any form of collaboration. So as you can see, we already established the licensing or partnership with different well-known companies, and we hope that we can accelerate the drug innovation through the external collaboration and partnerships. So please contact us if you are interested in any of them. So yeah, that's the last one. First, finally, I want to thank everyone for attending this webinar, and also I'm very happy to take any questions. Okay.
Thank you, Dr. Guo, for that very exciting and informative presentation. It looks like we have a few questions coming in from the audience. So it looks like, we introduced several different types of screening methods: beacon, phage display, and hybridoma. So could you comment on the advantages or disadvantages for those different screening methods?
Yeah, we know that there are different antibody screening methods, and the hybridoma is the most traditional one, which is very economic, but it's with low capacity and limited access to the B cell repertoire. The hybridoma is the widely used in vitro screening method, but it's also time consuming. And also, sometimes you can find the antibodies clones using the phage display has some issues for the CMC development, and also may generate high ADA response. But phage can use the native human sequences libraries to do the screening. That's their advantages. And the beacon that we used most often, because that is a high throughput technology, single B cell, which can identify the functional antibodies very quick within 1 day. That will save you 2 to 3 months when you use hybridoma or phage display. but that require a special machine. That's quite expensive. However, we already have that, so that's not an issue for us. So comparing all these technologies, I will prefer to use Beacon first. However, for some most difficult drug targets like GPCR, we would recommend you to use a combination of different screening methods to try to maximize the number of clones that you can obtain.
Got it. Thank you very much. So which method, so can target knockout efficiently increase the positive clone number? And can you let us know how much is that increase?
Yeah, I think I showed the data already in the slide that we use the target knockout mice for the immunization, and we try different drug targets. We see there is an increase about 2 to 10-fold or even more when you use the target knockout mice. And besides the number of antibodies that we can generate, the most important that we can generate antibody recognizing the conserved region between human and mice, which probably the most important functional regions. And also you can generate antibody recognizing different species that is more convenient for you for the in vivo efficacy test or the toxicity test.
Okay, thank you. So could you comment on which immunization method gives the best yields? I think you mentioned DNA immunization, but how would you compare that versus VLP?
Yeah, as I said, we prefer to use DNA because it's more economic. You can also it's very easy to use that to formulate. You just need to deliver the DNA into the mice using the electroporation, and that can generate proteins in its native structure and also with a long-term expression. The VLP we cannot make by ourselves, and also we know that it's relatively difficult to formulate. And also, although we compared the DNA and the commercial available VLP in some GPCR programs, we see the DNA, most of the time, both of them work, but DNA generate higher yields.
Okay. Thank you. So what criteria are we using to select lead candidates?
Yeah, that's a good question. So we have a lot of antibody sequences that we can generate after immunization. So initially we will do the basic in vitro assays, for example, the binding ability to its target, and also the species cross-reactivity, and also the family member cross-reactive test. And also we'll look at the binding affinity. And for GPCR, we'll basically look at the binding EC50. And for some case, we'll look at its function, like as an agonist or antagonist. If you are trying to generate ADC drug, we'll look at the internalization. Just based on all these in vitro assays, we will first of all to select some top candidates. Then we'll look at its physical chemical properties, the stability test, and also the in vivo test to finally end up with the PCC molecule. So that's do need a lot of work to finally to select your top candidate.
Okay, thank you. Next question. What is the chance to see antibody clones targeting an inaccessible epitope on GPCRs following your current immunization scheme?
What is the chance? So our strategy is to knock out the target gene first, so that will increase the chance. But I cannot tell how much increase it will. But compared to the wild type mice, we only generate antibodies with limited numbers and only recognizing the human antigen, not other species. We can say that the knockout mice will generate antibody with more diverse epitopes, and they can recognizing even novel epitopes.
Okay, thank you. Next question. Have we tried GPCR antibody discovery using the Ren nano model? And would there be any advantages to making anti-GPCR nanobodies over monoclonals?
Yeah, so we does have we do have a nano mice which can generate nanobodies, but that platform launched just early this year, so we haven't tried too many drug targets yet. But we have did some antibody discovery for GPCR proteins, and they are pretty successful. But if you compare to monoclonal antibody, we all know that the nanobody has a smaller size, which can penetrate better into the tissues. And also, in general, the nanobody has a longer CDR3 regions, which can reach a deeper epitope inside the GPCR, so which can facilitate its antibody function. So based on that, we think nanobody has their advantages. However, a nanobody has shorter half-life. That need to be considered when you're using it.
So the next question is about collaborations with academia. I think I can comment that, we are open for collaborations, with our Renmice models. I don't know if Dr. Guo, if you have anything to add about the collaboration opportunities, or if we can invite people to contact our business team directly.
Yeah, but if you're interested in that, please contact our BD team. I think we are open for the collaboration with the academic team, academic facilities. Yeah.
Okay, thanks. Let's see. So have we explored any GPCR targets related to metabolic disease? And what kind of results do we have?
Yeah, we tried some antibody discovery for the GPCR targets in the metabolic disease field, like obesity, diabetes, and NASH. So for example, we generate GLP-1R antibody that treating the diabetes and obesity, and we do see a good in vitro and in vivo function. And also we have a CCR8 antibody, which can be used for the NASH and also the liver disease. And we also see a comparable results to the benchmarks.
Okay, thank you. So I think there's a question about which GPCRs have you made target humanized mice, and where can we get the target list? Yeah, I can definitely, if you head to our website at biocytogen.com, you can download the full catalog that includes an extensive category of GPCR target humanized mice. And so I think we have a few questions for Dr. Kanke. So the first one is, what are the advantages of your membrane protein synthesis technology compared with others out there?
Okay, thank you for asking me. And so, well, first of all, so I'm very pleased to see, our antigen, membrane antigen worked very well for the CCR8 project, as Dr. Guo mentioned. And I just want to show you with a couple of slides quickly with our antigen technology. We have time. And at LiberoThera, we have proprietary cell-free membrane protein synthesis technology. With this technology, we can synthesize membrane proteins with appropriate folding. And so this technology allows us to make membrane proteins with integrated structure rather than disrupting its structure by extracting from cells. We also have additional technologies for non-canonical amino acid incorporation technology, which allow us to further modify the membrane protein adapted to the structure of the target. And this technology, I think another advantage of our technology is the flexibility and wide applicability of the many different types of GPCRs. We have so far tried to synthesize 300 GPCRs, and the synthesis itself works very well. But most importantly, we have to think about the structural and functional feature of each GPCR, because every GPCR has different characters, and we have to adapt to the target one by one. But so far, and so as shown in the CCR8 project, so our antigen resulted in very unique epitope of the antibodies. So structurally recognizing with discontinued epitopes. So that is a strong evidence our antigen technology has very good and also synergetic effect with the Biocytogen's technology.
Thank you, Dr. Kanke, for that detailed answer. I think this may answer some of the other questions. Can you comment on whether, so it looks like you commented on which GPCRs you've made using the cell-free protein.
Yeah.
See, and we have a number of questions and we may not be able to get to them all today. So let's see. I think back to Dr. Guo, what is our approach for targets where the knockout of the gene causes lethality?
Oh, yeah, I didn't mention this part. So we prefer to use the target gene knockout mice unless it's embryonic lethal. But if it's not embryonic lethal, it cannot do the knockout first. But you can optimize the immunization conditions, like adding different like the epitopes that can induce the CD4 cell recognizing and enhance immune response. And also you can add some different things in the solutions of the antigen to further enhance to break the tolerance, immune tolerance. So you can try different other methods. Yeah, if it's lethal.
Thank you. So we have a couple of remaining questions about Beacon. So how many positive hits do we typically observe, and are we able to run any functional screening?
Typically we run 4 chips each time using the Beacon machine, and the number of positive clones variable from different programs. Some we can get over hundreds or even 1,000 positive clones. We cannot export all of them because it takes a long time for the machine to export each single B cell. So we generally just export some of them. But basically we can see there are a lot of more potential positive B cells that can be screened if you have enough time to run the machine. For the functional assays, I think the Beacon system can not only testing the binding ability of those antibodies, for example, to the human antigens, to the mouse antigens and other species. It can also do some functional assays like blocking assays. But we normally just use that for the initial screening. Then once we obtain the sequences, we will make the antibody either in the CHO cell supernatant or in the purified proteins, then test the functional later on. But for the machine itself, it can do the functional assays.
I see. Okay, thank you. So it looks like we have one final question. Any comments on how, we can boost CD4 T cell responses to GPCR antigens? Any comments on that question?
Right, I don't have this question yet.
It's
Yeah, we will think about it and get back to that question later, or maybe by email. Yeah.
Okay. Yeah, please don't hesitate to reach out to us by email. We're happy to engage or answer any additional questions. It looks like we're out of time for today's webinar. Again, I'd really like to thank Dr. Guo, Dr. Kanke for joining us. It's very late where they are, so we really appreciate joining this live webinar. And with that, I'd like to thank our attendees for joining. We will reach out with a recording of today's presentation if you'd like to view it on demand. So stay tuned for that information. And in the meantime, again, if you have any questions, please visit us at biocytogen.com. You can contact us there, download our catalog, or engage with our scientists directly via email. So with that, I'd like to close today's webinar. Again, thanks to our speaker and panelists, and have a great rest of the day.